All right. Well, thank you very much, James, and the organizers for giving me an opportunity to talk about our work. This talk is really about how a group of inhibitors, a group of compounds called Integrase Strand Transfer Inhibitors bind to HIV integration complexes, which I'll introduce. And this is part of a long-term effort with collaborators of mine at the NIH to understand these integration assemblies and also to develop next generation compounds, next generation inhibitors. So my lab works on HIV. HIV is a lentivirus, which is part of the retroviral superfamily. So let's start with a little bit about the viral life cycle. So these over here, these are the early events that are displayed on this slide. Okay, there's a fusion event which releases the conical capsid core. Now inside the capsid core, there's a reverse transcription event which converts the viral RNA into complementary cDNA. After completion of reverse transcription and synthesis of viral cDNA, a multimer of integrase will then bind to the ends of the vDNA to form what's known as a pre-integration complex, or a PIC. And once in the nucleus, the PIC can then scan target chromatin and identify select regions for catalytic integration. Integration then really establishes a permanent and irreversible infection in the target cell. So this is integration as well as reverse transcription. These are really the two hallmarks of all retroviruses, including HIV. So we're specifically interested in the steps involving the PIC. Okay, the PIC is a large and it's still a rather enigmatic assembly. There's about 10,000 base pairs of DNA. There's viral integrase in there. There are cellular host factors. There's probably some capsid protein and unknown other host factors. It's basically a big messy assembly, and it really hasn't been amenable to homogeneous isolation for structural studies. Okay, but what we can do is we can assemble complexes that contain the basic functional components of a PIC biochemically, and then use them for structural biology. So what we do is we have oligonucleotides of viral, so about 25, 27 base pair oligonucleotides. So these represent the viral DNA. We incubate with integrase, and integrase can be a multimer. And under specific biochemical conditions, these will form into a complex that we refer to as a stable synaptic complex. Okay, integrase will then cleave off two of the nucleotides from the 3' ends to form the cleaved synaptic complex. And then if you add more nucleotides, so in this case, target DNA, then this cleaved synaptic complex will bind target DNA to form the target capture complex. And then integrase will perform its second catalytic event to form the strand transfer complex, which is the product of irreversible integration. So all of these complexes, for all of these complexes, we broadly as structural biologists refer to them as intasomes. Now, what's important for the rest of this talk is that there's a group of drugs called INSTIs, Integrase Strand Transfer Inhibitors, which target intasomes specifically. And these drugs are shown on this slide. Raltegravir and elvitegravir, these are the first generation compounds. They're clinically approved to treat HIV infected individuals. And as well as dolutegravir and bictegravir, which are also clinically approved. So these are the second generation drugs. Now these compounds, or these drugs, these bind intasomes and not free integrase. And I'll show you why that is in a later slide. Okay, so these compounds, they work here. They work by blocking the formation of the target capture complex intasome, and as such, they bind to the cleaved synaptic complex intasome. So these are intasomes with recessed 3' ends. Okay, so for our work that I will describe, we used 4 particular compounds. Bictegravir, this is the leading clinically approved drug to treat HIV-infected individuals. This really is arguably one of the best drugs in the clinic. It has a very high barrier to resistance. It's well tolerated. And these compounds, these are developmental compounds. They are based on a distinct scaffold, it's a naphthyridine scaffold, and these compounds are developed by our collaborators at the NIH, Terence Burke, as well as Steve Hughes. So we can purify cleaved synaptic complex intasomes, both in their apo form as well as bound to INSTIs. It's a multi-step purification. Actually, we get initially a rather heterogeneous assembly, a mixture of intasomes and then these higher order aggregates. We take them through multiple rounds of size exclusion chromatography. We actually use these aggregate columns to remove some of these impurities. And in the end, we get this peak right here off of a size exclusion column. It's not the cleanest peak, but it actually it works for our purposes. And then if you incubate, so you can show that these intasomes are active. So they will perform multiple rounds of concerted integration, and then if you incubate with any of the compounds, they effectively inhibit a block integration. This is what the data looks like. So we use cryo-electron microscopy to collect the data and determine the structures of these complexes. This is what it looks like. It's a little bit messy, a little bit heterogeneous. We actually had to use a number of tricks. We use these complexes have to be maintained at high salt, some amount of glycerol. If we also have to tilt the stage, so they're pathologically preferentially oriented, so we have to tilt the stage to run iterative classifications and so forth. Okay, so for those of you who are familiar with the methodology, so we introduced this idea of tilting to address preferred orientation a number of years ago. These are the types of reconstructions that we get in the absence of tilts. So they're very anisotropic, and that's just simply because the specimens, they're adhered in one orientation to the air-water interface. But then if you tilt the stage, then you can get much more isotropic reconstructions. Okay. So this is what the reconstructions look like within the conserved intasome core. And as you can see, these are twofold symmetric complexes, and we have 2 INSTIs, 2 Integrase Strand Transfer Inhibitors bound to the active site of these complexes. And this is just the density inside. So in the active site, so here I'm displaying 5 maps, one so the apo form, and then with the 4 compounds bound, bictegravir, as well as the developmental compounds, we could clearly resolve the metal ions. These are calciums in the case of calcium ions in the case of the apo form, and otherwise magnesium ions in the case of the drug-bound forms. And here's just a movie showing the density around the active site. So again, this is the drug. This is our leading developmental compound 4D. And these are the 2 magnesium ions. And then throughout the active site, we see many water molecules. Steve spent quite a bit of time explaining the relevance of water molecules in the context of connexin 4650, and I just want to highlight and emphasize that I mean, this is he mentioned these are like the 21st amino acid, and I couldn't agree more. They are so important. They are important in the context of enzyme function, and also in our context, they're also important for the purposes of drug design. So this is how the compounds bind to the active site. Over here, what I'm showing is the leading clinically used drug, bictegravir, bound to the intasome active site. You can see that there are 3 electronegative atoms that chelate two of the magnesium ions bound to the integrase active site. And there's a linker region that connects the core of the compound to a halogenated benzyl moiety. This halogenated benzyl moiety stacks on one of the bases of vDNA, and that particular stacking interaction between the halogenated benzyl moiety and the vDNA base is precisely why these compounds selectively bind to intasomes, but not free integrase. So the DNA is actually part of the active site. And then over here on the left-hand side, these are areas or regions of the molecules that confer potency, especially against viral resistant variants. The naphthyridine compounds, they bind closer to the 2 magnesiums, and that's just simply because the electronegative atoms are now embedded into the scaffold. So you can see that they're kind of pushed closer to the magnesium ions. You have the same stacking interaction between the halogenated benzyl moiety and the vDNA. And then over here at the 6th position, this is a region of the naphthyridine compounds that we are actively working on and modifying to make better binders, in particularly against viral resistant variants. Now, I do want to make a point that at a high level, these general binding modes have actually been known, and that's because we've had structures of prototype foamy-virus intasomes for about 10 years now. So PFV is a simpler retrovirus, and this is a distinct type of intasome. And the group of Peter Cherepanov showed a number of years ago now how some of these compounds, not the latest generation ones, but some of these compounds bind to the PFV intasomes. These binding modes are highly conserved. Okay, the problem is that the active site of HIV and PFV intasomes, and in general, the integrase enzyme, particularly the active site, they're not conserved. So depending on how you align the 2 integrases, the conservation of the catalytic core domain is around 20 to 30%, and about 40% of conservation around the active site. And this is really important, and it's important because when you overlay the 2 active sites, they're not identical. So this figure shows the RMSD between the prototype foamy-virus intasome active site and the HIV intasome active site, and it's mapped onto the structure of the latter. And what one sees is that while much of the active site is similar, or some of the active site is similar, much of it is actually distinct, especially in these bottom regions. And all of these residues here, these are viral resistant variants that are arising in the clinic. And what I want to emphasize is that all of these viral resistant variants, they are not conserved between the 2 retroviral species. So the consequences of this lack of conservation are actually rather profound. If we take one particular compound, this is the compound 4F. So this compound has a sulfhydrylbenzene extension on the left side of the active site at its R1 substituent. Okay, and we look at how it binds to the HIV intasome. What we see is that this particular chemical group extends out into the solvent exposed cleft. If we look at how that same exact compound binds to the PFV intasome, that sulfhydryl group actually bends back and makes an intramolecular stacking interaction with the core, with the naphthalene core. If we look at the compound 4C, which is really one of the leading developmental compounds, we see a very similar story. In the case of HIV, the pentanol extends into the solvent-exposed cleft, whereas in the case of PFV, it bends back. And so this is just a slide summarizing those findings. So this is with 4F, then this is with 4C, and I've also shown that the binding mode of the compound 4D to HIV is also conserved. So what this is really telling us is that if we want to understand how these ligands bind, if we want to utilize the information for drug design or for understanding mechanisms of viral resistance, we really have to be using the correct construct. This is absolutely essential. Now, I've mentioned that the residues in the active site are not conserved. And we know that drug resistance is a major problem. This is a snapshot, a table, actually part of a larger table. This is just one of the snapshots of the emergent drug-resistant variants against one of the leading compounds used in the clinic, and there's many, many more. Okay, and so what our future work is really focused on is to understand how viral resistant variants affect INSTI binding, and also how to utilize that information to make a better drug, to make a better compound that will work against drug-resistant variants. And with that in mind, I want to finish up on this one slide. So this slide describes the general strategy that we are using, that we're using to achieve this goal. So it's based on this idea called the substrate envelope hypothesis. It's actually an idea that was initially developed by Celia Schiffer's lab and applied to the development of protease inhibitors. So it describes the idea of the substrate envelope. The substrate envelope is a molecular envelope of an enzyme that binds natural substrates, okay, any natural substrates. In the case of protease, these are amino acids that are proteolytically cleaved by the enzyme, and in the case of integrase, these are both viral and target DNA. And so basically the idea is that a resistant variant needs to displace the bound drug. And if a resistant mutation arises, it will, so if a resistant variant arises and it displaces the drug while at the same time protruding into the natural substrate envelope, it will compromise viral fitness. Okay, so if you keep the drug within the substrate envelope, then a resistant variant will also compromise viral fitness. But then if the drug protrudes outside of the substrate envelope, then a resistant variant can much more easily displace the compound without actually compromising viral fitness. And so therefore what we're doing is, as we're optimizing these compounds, we're now really using this idea of the HIV substrate envelope to guide us and to make sure that we're constraining the molecule within its boundaries. And so with that, I'll just conclude. So we now have workflows for intasome inhibition and structural studies. We have sub-3 angstrom resolution, which is providing insights into the structural differences between HIV and PFV intasomes, drug binding, the role of solvent. Showed that using the correct specimen now for understanding how these compounds bind is essential, and we're using the substrate envelope hypothesis to guide future structure-based drug design. And just to finish up, these are the people who did the work. This is Dario Passos. He's a fantastic staff scientist in my group, along with a postdoctoral associate, Lorna Joswick, who did the structural biology modeling. This has been a long-standing collaboration with the group of Bob Craigie and Min Lee, at the NIH, and we have a great group of collaborators at the NIH and at Scripps, and microscopy and computational support. And thank you for your attention.